This video abstract is AI generated. How can two enzymes with nearly identical binding sites recognize the same ligand so differently? In this work, we studied the C-terminal domain of bacterial enzyme 1, or E1C. This domain binds phosphoenolpyruvate, PEP, and initiates the phosphoryl transfer reaction that powers the bacterial phosphotransferase system. E1C is also an attractive antimicrobial target, but designing selective inhibitors is difficult because many enzymes and metabolites interact with molecules that resemble PEP. The central question is therefore, what controls ligand selectivity? We compared mesophilic E1C from E. coli with thermophilic E1C from Thermoanaerobacter tencongensis. These enzymes share the same overall fold, conserved PEP binding residues, and similar affinity for the physiological substrate PEP. Yet two non-hydrolyzable PEP analogues, SEP and CPEP, behave very differently. NMR chemical shift perturbation experiments showed that both SEP and CPEP strongly perturb thermophilic E1C, with changes localized near the PEP binding site. In contrast, the same analogues produce weak or negligible perturbations in mesophilic E1C. The analogues selectively recognize the thermophilic enzyme. Hybrid enzymes then revealed where this selectivity comes from. When mesophilic binding site loops were placed onto the thermophilic scaffold, analog binding was weakened but still retained. But constructs with the mesophilic scaffold showed weak or negligible binding. This indicates that the protein scaffold, not only the local active site loops, plays a major role. Molecular dynamics simulations explained the mechanism. PEP sits near the center of the arginine-rich binding pocket and forms stabilizing hydrogen bonds with backbone amides. SEP shifts toward arginine 296 and arginine 332. Weakening these stabilizing contacts. Combined PC of the simulations revealed the key dynamic difference. In mesophilic E1C, SEP binding leaves a residual breathing motion of the barrel. This motion expands the binding pocket and destabilizes the ligand complex. In thermophilic E1C, the scaffold is more rigid. The breathing motion is quenched, and SEP remains bound. The final model is that ligand selectivity is encoded not only in the static binding site, but also in the motions that remain after binding. Protein conformational dynamics can therefore be exploited to design inhibitors with subfamily selectivity. To know more, read our work online.